Okay, so welcome to the Society for Ecological Restoration Northwest Chapter Missoula Pub Talk. Tonight we have Scott Robinson here with us. He's with the Ecology Lab at the University of Montana. And Scott has a Bachelor of Science and Master's of Science in Forest Ecology and Management. And he is currently getting close to finishing up his PhD studying or research on metal contaminated soils. So thank you very much for taking the time to come talk to us, and we're excited to hear about the research.
All right, thank you. Thanks all for coming out tonight. I know it's a busy time of year with spring being here, field work, getting ready for field work and yard work and whatnot, places. So tonight I'll talk about nitrogen and phosphorus limiting plant growth in a smelter aerosol-affected soil near Anaconda. So I'll start by briefly introducing the topic, background on what nutrient is, then I'll introduce our sort of project site and talk about some preliminary results and hopefully with maybe some insight into what we found or just some good talking points for future discussion maybe.
No, just click on this right here, just the right mouse.
Right.
Yeah, just right here. Oh, it's working now.
Okay, so this idea of nutrient limitation or a growth limiting factor, something that limits growth, is not a new thing. In fact, it came out in the 1830s. It's formerly known as the Liebig's Law. And what it says is just that plant growth can never be more than the level allowed by the growth factor present in the growth. And this is often represented here in what they call the Liebig barrel. And this example here is with phosphorus. So phosphorus is the lowest state making up this barrel. And in this agricultural example, the yield in whatever crop that is can never exceed the amount of phosphorus that's found in the soil present. Now, other things can be, I have a lot. Other things can be, other factors can be limited, but it's generally nutrients, especially nitrogen and phosphorus, most common, at least in what we study and what we find. This work by Liebig also gave rise to the invention of nitrogen fertilizers and fertilizer use in agriculture. And implicit in this sort of Liebig law is that you cannot add something that isn't limited to see a response in growth. In fact, if you start adding things that aren't limited, you're going to throw that system out of whack. And that's exactly what we see today with modern. Since 1960, the use of nitrogen fertilizer and peat, this goes beyond 1960, but it's sort of out of control. We're adding it because it's cheap, it's cheap, it's easy to apply, and so why not add it? But what this has done is this has ultimately changed the global cycle. And most of the nutrient limitation studies that you see out there really are framed where they're looking at the change in these global cycles. I will not be talking about that. I'll be talking about metal contaminants, but I just wanted to point that out that our use of fertilizer has totally changed. So more recently, this idea of nutrient limitation has occurred in ecosystem ecology. It's helped us understand how ecosystems are structured and function. And using this example here of this forested ecosystem kind of changing over time, becoming more complex, what we know is that, or in general, what we think is that phosphorus is at its most available early on. It is sourced from the rock, the parent material that makes that soil. And that nitrogen at this point, early stage, is mostly biological. So inputs of nitrogen into the system are mostly through nitrogen fixing. So in this case, again, in this simple general model here, we know then where we think that early on in the successional stage, these systems are nitrogen limited, with the bulk of the nitrogen coming in being fixed. That nitrogen is eventually built over the system and no longer becomes nitrogen limited. However, as this soil is developing and this ecosystem is developing, becoming more complex, aging, going young, 100 years to 100,000 years, becoming young to mature, that P is becoming included in the soil, or it's becoming mostly in biomass. And so we have a switch from a young soil or a young ecosystem that we think is nitrogen limited to an older system, a mature soil that we now think is phosphorus limited. We can extrapolate that then to just disturbance or looking at age. Again, we can think that, like, say, for Montana here, these are relatively young since the glacial period. So we have young ecosystems. We can think then that generally these high latitude are nitrogen limited. But tropics or warm climates where there's been a lot of, say, soil, where these ecosystems are now more mature, these are P limited. So nitrogen limited, where we live, phosphorus limited, tropics. Of course, it's pretty simple. It's a good starting point, but it's pretty simple. And we know now that it's actually a diverse set of factors that influence nutrient availability. And in fact, one major one is, again, this parent material. So that last example I was using, looked at unweathered rock. So something like paddy in Montana. But also in Montana, we have these lush soils over by Bozeman, which is just wind-derived material. A lot of that stemming from after the glaciers receded, before vegetation, but also the dust bowl blew a lot of this wind-borne material. Well, generally, we see that these soils are producing, or grassland vegetation is really much predominant on these lush soils. So what we can say then about parent material is that this loess is going to have a different amount of P available, say, than this unweathered rock. And based on climate, that P is either going to stay longer, or it's going to be removed quicker, or occluded, or taken up in biomass based on how wet it is, how warm it is. And also vegetation is a big player in controlling sort of nutrient limitation itself, because it is a big proponent of how the soil itself develops. And we can expect, again, with this Bozeman example of loess and grassland vegetation, that we see a whole lot of calcium carbonate here, and grasses are considered to be, at least when I was taught, base pumpers. So we can expect there to be a naturally higher pH, maybe the pH that's more favorable to having these nutrients available, say, than a lodgepole pine stand that was formed over sandstone, which would be sandy, acidic, not much texture to it, and probably poorly nutrient position. In fact, going further, there's been now enough data and enough of these sort of fertilization studies where they add N and P, or N plus P, to where we have enough data out there to talk more broadly about nutrient limitation in terrestrial, freshwater, and marine ecosystems. And the big one that came out was by Jim Elser and friends. He's up at Applied Biology Station now. And they did this huge meta-analysis looking at these different fertilization studies. And what they found is that in most all ecosystems, terrestrial, freshwater, marine, there is a response to N being added, so they are nitrogen. Well, they also are responding to P, so whoa, also phosphorus. But they're really responding to the addition of both N and P, which makes them potentially co-limited by both phosphorus and nitrogen. What they also found in this analysis is there was no latitudinal gradient. So this idea that our soils up north here have more P than, say, a soil that's heavily weathered in the tropics, that did not show up with their measurements. Fast forwarding to this Fay et al. Paper, which is part of this nutrient network. We have a site, Elva, at MTG Ranch, and what I'll be talking about, we kind of borrowed some methods from that. But they set up this sort of global experiment where they were adding N and P alone and in combination with other nutrients on a global grassland scale. And what they found was that productivity was co-limited by multiple nutrients, similar to what we found here, or not we, but also found here, was that terrestrial ecosystems appear to be limited by both N and P. However, I would like to say that this study also did happen to find a gradient in which nitrogen limitation peaked at northern latitudes. So there's evidence to suggest that what we just talked about still holds some truth to that. So it's a good starting point, right? It's a good way to sum up your question. So what we're really interested in, or what I'm really interested in, based on mining, is what happens after large-scale disturbance. And this could be any disturbance, it could be fire. In my case, it's mining or smelting. And I want to know what happens after a major disturbance removes its vegetation. Well, we can think then maybe that it might get set back. If this is a young system like we have here in Montana, then your vegetation is just gonna move back to successional. You haven't developed that much soil, you haven't lost that much. These mountains are still quite large, right? So we would think that it would just start over, maybe with nitrogen fixers coming back. You still have a plenty amount of P, phosphorus available to promote this ecosystem development. But on contaminated sites, that becomes a whole different story. Like, we don't really know what the responsive nutrient fixes are to metal contamination. Are they going to be there to bring nitrogen into that system, or can we assume that they'll just probably be nitrogen limited? But also, just in plant growth in general, these high energetic costs associated with dealing with metals, that's going to cost a lot more nutrients. So maybe even if there is nitrogen fixers in nitrogen and P's and plenty of stuff, maybe that's not enough to help these plants get over that contamination to help them establish new growth. So to formalize these questions a little more and get into the study, we wanted to know, based on this background that I just gave, is nitrogen, because we're near Anaconda, so high latitude, young soil. Is it limiting, or is it a combination of nutrients, P plus micronutrients or NEP, that are limiting plant growth in this metal contaminated soil? And then also, do these different species, do different plant species respond differently, or do they have different nutrient needs to establish in these metal contaminated soils? And then lastly, what, if any, are there romantic implications to help restoration move forward, maybe selecting for which fertilizers to use, or which fertilizer is important when, and hopefully this will get into that. Hopefully this study will have some insight into that. So this is one of the sites. This is a common. This is all paper my business work. Sure, most people that. So this is one of the things that he's doing on these steep slopes. He's adding this map here, and then he's trenching and adding fertilizer and seed to try and, to one, establish vegetation to stabilize. Establish vegetation to stabilize the slopes. But what we can see here is there's some. This is after 2 years. There's some serious spottiness to this that sort of begs the question, why is this fertilizer was applied here, not just here? So why is it there here, and why is it growing there? Or even so, why is it not growing in between? So that is some of the stuff that we're trying to do with our work, and maybe this study will help add to that. But where else, like I said, I just pointed that out, so it's kind of cool. But where else is there a better place to study mine contamination than in our own backyard and upper part fork here? So we have a 150-year plus mining legacy here in the Blackwater, so rich history. I'm not so much worried about this talk, mining itself, but actually Anaconda and its great smelter here. And specifically, I will be talking about this area south of Anaconda, called Mount Hagen Uplands, which is a small portion of the larger Mount Hagen wildlife. So if you zoom in a little more on this Anaconda smelter site, Anaconda being here, here's a Slutty Mountain, Mountaineer Ponds, and just south of Anaconda, 4 miles or so, air miles, maybe not even 2, is this area known as the Mount Hagen. And this area here should look like that, but it does not. So it's been polluted with aerial emissions from the smelter for about 100 years. So again, Mount Hagen Uplands was receiving about 100 years of smelter airborne emission inputs from the Washoe smelter stack. This is 1910. Sorry, changed picture and changed that. But what you can see is the smelter here, and that is not clouds. That is airborne emission from the smelter. And around that time, 1910, they estimated that 5 to 8 miles around that smelter, all trees. So it was releasing upwards of 30 tons of arsenic and lead and all that good stuff a day. So these huge and long-term inputs of contaminants coupled with these large timber sales, the largest timber sales in the country at the time, this is intentional. They ran this log flume up and over the Continental Divide all the way to the substation outside of Anaconda, so 20 miles. Just really long flume. So these two things coupled together. Now this is 39 years post smelter cessation, but this is pretty common of what the landscape looks like in the Mount Hagen area, if you're not familiar. Where water is moving, we have trees. Where the aspect is favorable, we have vegetation. Where the aspect is not favorable, we have bears, lots of bears around, and lots of erosion. And what this comes out to be is about 9 square miles or 5,500 acres of injured area in Mount Hagen. So quite a large area. So our study site for this project, which I'll get into, is up here on this, in these ridges up here. And what we did is in 2018, we went out in April and May and we found the site that is fairly contaminated for that area. We're talking about 4 years of erosion, so metals aren't as high as they used to be, but they're still high enough. And so we picked the site and we found this little runway here. And what that looks like is it's 5 blocks, 10 plots. And each plot here is 1.5 meters by 1.5 meters, 2.25 square meters. This whole 2.25 square meters received some combination or one of these treatments. So we have a control, which was nothing was done. We have a seed only, which was a native mix of 3 trees, 3 forbs, and 3 grasses, all at 10% of the seed mix, so equal percentage seed mix. Then we added potassium and phosphorus, nitrogen alone, and then we coupled those up. And then we also had one where we added NP and K together. And the main form of nutrients being added out there during respiration are the main fertilizers, same fertilizers. So if it's one, it might hold more water. But 2, it is this organic N base, so it takes longer to break down as opposed to organic N types. But like I said, we had NPK and we added them alone or in some combination. And they were added at 10 grams of nutrient per meter squared. In 2018 or last spring, this whole area was raked clean before we counted all this rebar. So we really started from nothing. So we have 0% cover. So everything we see is fresh. And then we came out back in July, and we measured these at the end of July, and so I'll be presenting on one growing season data on a harsh site. So, yeah. But it's, you know, some trends are emerging. So, like I said, this plot here, right, is 2.25 square meters. That all received the fertilizer. And then we broke this up into a 1 meter squared plant cover plot, which will measure every growing season. And then in order to make this go 5 years, we have these biomass plots that are 0.2 meters squared, and we will clip these every year, every year, so we get 5 years out of that to get above ground net final based on it. And so, like I said, this is set to go 5 years. It'd be nice to do it longer, but that's a lot of rebar to carry up there, and we also kind of bring up space. So it'd also be nice to replicate this on a soil type too, but as of right now, we just have it on the site. Just briefly talk about the results. I'll only present today on total cover. We're still working on the biomass stuff. It's not quite as a clear story yet, and I think it really has something to do with just how we're fertilizing and raking. Just didn't get spread out in these. Anyway, I'm going to present on the cover data, and for this first plot and every other plot, we'll have cover on the Y, and then we'll go control, the seed only, which is just that native seed mix, potassium alone, phosphorus alone, nitrogen alone, potassium plus phosphorus, nitrogen plus potassium, nitrogen phosphorus, potassium, and then sustain on the end. And so if you see these letters, this A and this AB, this means they're not C. This C, this B, this C here, these three, so our NMP, our NPK, and our sustain treatments are all significantly different than the control, meaning then that these treatments resulted in more plant cover with the addition of those fertilizer combinations than the control being left to its own. At least with NNP and sustain, these two produced more plant cover than the seed only. The potassium treatment was our worst performing treatment. That also, which I forgot to mention, also had an addition of micronutrients added with it, a one-time addition of micronutrients. This sort of micro mix, this pulse of, we had more copper, zinc, magnesium, all the different micronutrients. And so I have this feeling that addition of water-soluble copper might have been actually driving this. But it's only a one-time application, so hopefully that'll pick back up as we move on. But nonetheless, after one growing season, and like I said, this is total cover, so this is grass or tree. Really, this is grass. We had very little response to forbs, and the trees were so small that so a better way, I think, the tree average count. But I'm not going to get into that here. But I think there's pretty convincing evidence after 2 months, after one growing season, that there is definitely a multi-element limitation on getting these plants established relative to businesses usual, so to speak, this control.
What were the nutrient levels in this? Did you test the soil before, the existing soil before you added nutrients? Were there any nutrients in that soil at all?
So I don't have that data right now for this. I have nitrogen. So we can't really detect, at least maybe it's a method thing, we can't really detect nitrate.
Even after you added it?
Well, we don't have that. But starting out, it's very low in NH4.
What is in that soil other than that? Any organic matter?
Pedro has,
Oh, Peter's here. Hi.
It's like Steve, I mean, mostly a Steve Horizon. For the
Just
most part, all the
bare.
fines were blown away and washed away.
no nutrients, no organic.
Yeah. And that side is relatively decent compared to some of the steeper slopes where we've lost 24 inches. So then we really just see.
but you have data that suggests that it's pretty rich in phosphorus.
Yeah. From the parent material itself. It's just it's in there. I don't know the availability. That I don't.
Right. And so we have that, we'll be getting that data. We just don't, we don't have.
And then another quick question.
Sure.
What's in the sustain?
The sustain is organic, it's turkey.
It's like it's still nitrogen,
It's
phosphorus,
8 to
just a different concentration.
yeah, much lower, but we hammered, so we put 3,000, be the equivalent of 3,000 pounds per acre. So it's a one-time pulse. We're walking away from it, and we're just going to measure it every year now to see what's the longevity of that one pulse sustained over time. Where these other elements, these other nutrients will be added 10 grams per liter per acre. So that sustain is it's organic, it's 13 crap, and it's 824.
It's like it's ground is like byproduct of after they process turkeys and grab everything else and mash it up. It smells like.
It's interesting because we have the nutrient box on the ranch too, and you know I've seen we have 3 sites, and I've seen consistent reductions when we're adding potassium as well. So it seems like not potassium by itself, but it seems like it's reducing the potential of nitrogen to plants to respond to nitrogen.
The cover is reducing when you add phosphorus?
Biomass, yeah, we're measuring biomass, and you know there's a consistent thing across all 3 grasslands that we're testing, and it's just like when you're adding nitrogen, they respond a lot because we had a wet spring last year, but you're adding potassium with the nitrogen, and it's a reduced growth. And I mean, it could very well be with the micronutrients, but we're not adding those together with potassium.
Yeah.
So I don't know what's going on there. So it's kind of interesting.
Sure.
You're not just seeing with the NK, you're seeing with the NPK.
Right.
So when we're adding that, you're adding reduction, but it's a pretty substantial reduction.
It is.
And I don't know what the potassium would do. That would be harmful. I'm asking the plant physiologists about that.
Robert Powell, Montana Tech, he has some data that suggests that it is actually important.
Yeah,
So,
that's interesting. Absolutely. I mean, it's interesting to see that you see them.
And you have real soil there.
Yeah, no, that's what we do. Yeah, we have not pretty healthy grass there. So, yeah.
So I'll move on now. Like I said, this is primarily just grass. And that's not huge cover, but like I said, we started at 0. And after 2 months up there, I'm pretty happy with what we saw. And so I'll just focus on grass and X those 2 out from below millimeters because we did see some actually differences in the response of the grass species. We planted Element Street Cross Sunny wheatgrass and Blue Bunch wheatgrass, and then we also had basin in our mix. And here's to me, the really interesting part is the bulk of that NPK that we saw driven down over in total that we just have been talking about actually produced the highest amount of native grass, more so than NP and sustain. And then again, we just really got nothing with the control or the seed only or these nutrients in this combination other than the NP, NPK. So this is very interesting to me. The bulk of that cover that we received that we got out of the NPK was actually native. Where the response of red top to NP and sustain was pretty much the whole of that original.
Oh, this is just fascinating.
You heard me in that conference a long time ago.
I mean, now I want to go fertilize all of Milltown with, get rid of the rest. That's interesting.
So, you go back then, so are you saying then that the response, the NMP and the sustain, the response that you're seeing is from the existing seed bed?
It has to be.
It has to be, right, so that growth
That's
is
the
basically
best way.
stimulating the existing seed bed versus that is just the seed mix, and it's what you put into the seed mix that's responding to that K.
Right.
Right.
So we,
So
So
it's like
that could
it's like
be-
a phase of
yeah.
growth. It's like that process is important for seed to seedling, but not important for existing. Maybe that.
so we tried to separate this out. We did our best to rate out that whole area. But some of it was right. We couldn't do everything.
Yeah.
Is
So
that the dominant species there?
the
Or-
is, yeah. So what we did then is we did our best guesstimate to look at what we thought was actually from seed based on the size, what we think would be 2 months of versus what was already out. And so we subtracted. So everything I'm presenting is our best guess at what came from seed.
That's the only species that came in from like a seed bank?
It's hard to tell what our, all these that we planted grass-wise, those grasses, those are essentially tree grasses, three predominant grasses out there. So I don't know if it's the seed bank or if it's seed mix. We're guessing it's from seed. So that's another. Is there evidence to suggest using seed mix? I would say no.
Where do you go with seed? Is it-
It's all from Montana.
Okay.
The native plants program. Yeah.
Any evidence of other invasives in the making headway?
No, we found bromus. Another plot from no.
Cheatgrass?
Right
in the pot.
In
Is that
a
not
lot
up there
in the
yet?
next watershed over, but as far as this site, it's really so not too bad. There is on the hills, on the slopes, there's as far as in our plots. All right, and then so yep, the grass is sitting. Again, we saw the response. And the sustain, as opposed to the natives, which preferred the NPK. It was the best performing treatment combination for the natives. And so I would just like to bring this up because that's, to me, this is really the battle out there. I mean, we could have all the key in the world, but we're the only soils on pH 5. And so at pH 5, copper and zinc is pretty much at its maximum bioavailability. Iron is its at, in this soil, rich with iron too. I don't know if it's necessarily from the volcanic bedrock or if it's from the smelting process or a combination of both, but there is a lot of iron in that soil. And so at this pH, iron is going to be highly available as well. And what we know is that when iron's available and we have a low pH, that this iron really just binds. And so that probably is the important part or one of the main drivers of why we're seeing this response to P, even though there's steep, rich bedrock. Whatever iron there is available is binding this P. So with that, I would just conclude here, say that we did see an N plus P response that was greater than N alone, and P alone was also greater than the sum of those together, which makes it synergistically co-limiting. And we saw that different species appear to be responding differently to nutrient conditions. natives versus the grasses. And then the question that we've kind of just been talking about, or what people about, is this just an early response to the plant community? Do they need peat, say, to get started, and then over time nitrogen plays a bigger role? We don't know, but we set this up for 5 years, so it's not really long, but it's long enough to maybe at least with grasses pick up some of these responses. And again, you alluded to that too. That I would suggest the use of micronutrient fertilizer. At least there, and it's important to getting this vegetation started. We'll see what happens over the next 5 years. I'm putting it this weekend, so see if I can fertilize. No more.
Bring your skis.
Yes, there's skis and snow.
There's still California Creek is just the ice is just breaking. Literally just
We ain't
breaking.
seen nothing yet. It's been Outer Banks up there for like, it was Outer Banks for 10 days.
Really?
Yeah.
Yeah, no,
before
it's
April went
a
off.
completely different, it's like a month difference. As soon as you cross over the divide up there, it's like a month.
So are there any grasses you seeded that are those the only two native grasses you
We
seeded?
could
Or?
identify.
Yeah.
Just, weeks.
What other species were in your seed mix?
Basically,
Just those three, so
Yeah.
just based and then you didn't see them.
I couldn't tell you which just by looking at them. So we learned from this. So they do at least one of that's much different. The ground space. So you can tell this. So that's kind of good. And that's another thing that we do in this project. Since we learn is actually parsing this out. Yeah, that was good.
All right, I'm going to stop recording. Maybe.